So thank you again for inviting me as a resource person for this very important FDP. The topic is very important, AI and machine learning applications. What today I will talk about in this session, the topic is multi-objective genetic algorithm and applications. You see that most of the engineering problems can be posed as optimization problems. So that means given some decision variables, you have to optimize certain criteria to optimize certain parameters for a system. So here I will discuss multi-objective genetic algorithms. So a tool for optimization, multi-objective optimization. And then if the time permits, I will try to discuss some of the applications. I mainly work in computational biology domain, so I will prefer to give at least some glimpse of some applications in that domain. So I will prefer an interactive session, so at any point of time, if you want to interact, you are welcome. So first of all, what is multi-objective optimization? So you see that in real world domain, we have to deal with several optimization problems. So just for example, suppose I want to travel from Kolkata to Vizag. So we might have several options. One may prefer to travel by train. Someone may prefer to travel by flight. Someone may just want to drive, or even you can walk. And you want to optimize certain criteria, suppose. For example, you want to minimize the cost of traveling, and also you want to minimize the distance that you cover. So it might happen that if you want to, and also you want to minimize the time of traveling, that how much time it is taking. But the problem is that if you want to minimize the cost, you have to say the travel by train. But in that case, it will take more time than traveling by flight. But in that case, if you are traveling by flight, it will take more cost. It will cost you more. So these criteria, these, what we say, that objective functions that you want to optimize might be contradictory in nature. That if you want to minimize time, the cost will be more. If you want to minimize the cost, the time will be more. So then what solution we should take? That should I travel by train, or should I travel by flight, or we take any other option? So this multi-objective optimization deals with this kind of problems where you have to optimize multiple objective functions, and there might be also certain constraints. So here is another example. Suppose I want to purchase a car. You want to purchase a car. So there might be several objective functions to optimize. For example, you want to minimize the cost of the car that you were trying to buy. You want a minimum insurance premium, and also you want a small weight for the car. Whereas you also want to maximize the comfortability while in the car. And these criteria sometimes might be contradictory in nature. That if you want to optimize one, the other will not be optimized at the same time. And also there can be certain constraints for the optimization problem. That means you want to achieve all these optimization of these objective functions, but also you have to satisfy some constraints. Like you will choose only those cars that will have a good stereo system, that will seats for at least 6 adults, and it will give a mileage of 20 kilometers per liter. So these are the constants. So you cannot choose any other car which do not satisfy the constants. So you have to satisfy the constants as well as optimize these criteria. So, and what are the decision variables? Means the variables on which these objective functions are actually dependent on. So that might be the size of the car, that might be the length and width of the car, that might be the color of the car also, and many other parameters that actually determine these objective function values, on which these objective function values are actually dependent. So these are called as the decision variables. Hence, purchasing a car is a multi-objective optimization problem. So in many real-world problems, we have to simultaneously optimize multiple objective functions. And these objective functions are often contradictory in nature. As I already give an example, that if you want to optimize one criteria, the other criteria might be having very bad values. So obviously for these problems, the finding a single solution that I will definitely purchase this car. So if you have only one objective function, suppose your objective function is the cost of the car and you want to minimize, then your solution is easy. That means out of all cars, you choose that car that has the minimum cost. But when you have multiple objective functions, then there might be it will be very difficult to find a car that will give you the optimized value for all the objective functions. You might not get a single car which will have the minimum cost, maximum comfortability level, and say the other objective functions that it will having the minimum insurance premium, minimum weight. It might not happen. You may not get a single car that will optimize all the objective functions. So most of the cases, what you will get a set of solutions. Set of solutions means one car, you will get one car that will have the minimum cost. You will get another car that might have the minimum premium insurance. You might get another car that has the minimum weight, and so on. So you will get a set of solutions that when there are multiple objective functions that you are dealing with. So when we are dealing with multi-objective optimization problems, so you might choose to optimize each criteria separately. That is one option. But just optimizing, suppose you target the cost objective function and you want to minimize it separately. So formally, if we want to define what is a multi-objective function, values depend, on which the cost of the car will depend, on which the weight of the car will depend, and so it's less than that might have a lower cost, but you cannot choose it because it is outside, it is violating the constants. So there might be a set of constants, might be some inequality constants and also some equality constants. Equality constants means that it might have that you want a car that will exactly have 6 seats. So that is an equality constants. You don't want greater than 6, you also don't want less than 6, you want exactly 6. And inequality constants means you need at least 20 kmpl mileage. So you more than that is allowed, but less than that is not allowed. So for the optimization problems, you will have certain constants sometimes, and you are going to optimize a vector function. Vector function means again a set of objective functions. So one function is the cost of the car, another function is the weight of the car, another function is say the insurance premium for the car. So basically vector function means a set of functions that you are going to optimize. So this is the formal definition of multi-objective optimization problem. So you see that in multi-objective optimization problem, we have to deal with two search spaces. So one search space is the decision variable space or decision space. So suppose we have three decision variables: the length of the car, the weight, say the length of the car, the width of the car, and the height of the car. Say for simplicity, let us assume that we have these three decision variables. So each of these points is actually one car. So for example, for this point, this car has a length of this, width of this, and the height of this. So every car can be considered as a point in this decision space. Fine? Because every car will have length, width, and height. So we can plot the cars in this decision space. Similarly, we have an objective space. Suppose we are dealing with just two objective functions, the cost of the car and the insurance premium of the car. So so for example, for this car, we'll have certain cost and certain insurance premium value. So that car will also have a corresponding point in the objective space. If you are not getting me, just you can interrupt, okay? So every car has a point in the decision space as well as has a point in the objective space. Right? So corresponding to each car in the decision space, we will have a point in the objective space. Now the problem is that if two cars are very similar to each other in the decision space, that means very similar length, width, and height, that does not guarantee that they will be very close in the objective space as well. Even they have the similar variable values in the decision space, but that does not mean that they will be very close by in the objective space as well. That their cost and insurance premium might be very different. And vice versa. That means if two cars are very close in the objective space, that does not mean that they are very close in the decision space. So the optimization problem is that it is basically a searching problem. You are searching for a car in the decision space that will give you the optimized value of the objective functions. So that's why it is a difficult problem when you are dealing with the multi-objective optimization, when you are dealing with multiple objective functions. So formally, suppose you just forget about these mathematical notations. I just want to give you an idea what it is actually saying. Suppose we have two solutions, two cars. Here solutions means one car. Every car is a possible solution, and we are looking for a car that optimize all the objective functions. Now suppose you have only one objective function, that is a traditional single objective optimization problems. One objective function, that is cost. How do you compare two cars? How do you say that given a car A and car B, which car is a better car in terms of the objective function cost? Obviously the car with lower cost, because this is a minimization objective. The car with the lower cost is a better car in terms of the objective function. Better in the sense it is always in terms of the objective function, right? But if you have two objective functions, suppose one is the cost of the car, another is the insurance premium of the car, and again you are trying to compare two cars A and B, it will not be as straightforward as in the case of single objective problem. Why? Because it might happen that the first car, the car A, has a lower cost than car B. But car B has a lower insurance premium than car A. So which car is a better car? It is not straightforward. So if you get that car A has in both the objective function, it has lower value than car B, then definitely you say that car A is better than car B. Or car B has lower values of the objective functions, both the objective functions than car A, then you say that car B is a better car. But what will happen that one car is better in one objective function and the other car is better in the other objective function. Then you cannot say that the car A is better than car B or car B is better than car A. So here is an example, you see. Suppose we are comparing two solutions, solution A and solution B, and you have three objective functions. And I assume that all the objective functions are of minimization objective. That means you want to minimize all the objective functions. Because the objective functions might be either minimization objective or maximization objectives. But here I am assuming that all the objective functions are minimization. So for these situations, so this says that solution A or car A has an objective function value for f1, the value is 2. The objective function value f2 is 4 and f3 is 3. And for car B or solution B, the objective function values are 2, 7 and 6 for these three objective functions. So can you say that which solution is better, A or B? This is all of them are minimization objectives. Solution A is better because for f1 both are same, but for f2 and f3 A is better than B. So overall I can say that A is better than B. So in multi-objective terminology we say that solution A dominates solution B. Okay. On the other hand, for this case, you see that for f1 and f2 both are same, but for f3 B is better than A. So in this case B dominates A. In the third case, this is the problematic case actually. So here A is better in f2 and B is better in f3. For f1 they are same. So neither A dominates B nor B dominates A. So in this case we say that the solution A and B are non-dominating with each other. So in multi-objective optimization, when you have to compare two solutions, these three possible cases might arise. Either A dominates B or B dominates A, or they are non-dominating. Okay? So now you see that, let us see this picture. Suppose we have two objective functions. So this is the objective space. Two objective functions f1 and f2, and I assume that both the objective functions are minimization objectives. And these are the different solutions in the objective space. And why the space is having a shape like this, some uneven shape? This is because of the constraints. Because there might be some solution here, but that does not satisfy the constraint. So that cannot be considered. Now, if you compare any two solutions in terms of the objective functions here, just for example, if we compare, say, solution x1 and x7, can you tell me that what is the domination status between these two solutions, solution x1 and x7? Who has better value in f1? x1 has a better value in f1 because both are minimization objectives. And x7 has a better value in f2. Right? So x1 and x7 are non-dominating there. But if you consider suppose x3 and x7, here x3 is better in both the objective function values. Both in f1 and f2, x3 has a lower value than x7. So in this case, we say that x3 dominates x7. Similarly, x4 also dominates x7. Agree? x4 also dominates x7. It is very clear because if any solution is there within this box, then that solution will dominate x7. Because that solution will have lower value in both the objective functions. Now, out of these set of solutions, what we are searching actually? Which solution we are searching for? Optimal solutions, fine. But what is the definition of optimality here? In case of single objective case, definition of optimality is very easy. That means out of all possible solutions, we want the solution that gives the optimized value of the or minimum value of the objective function. But here we have two objective functions. Then what is the definition of optimality? Which solution will be called as optimal solutions? Here, the definition of optimality is like this, that out of all solutions in the search space, this is called the search space or the objective space, we are looking for those solutions which are not dominated by any other solution in the same search space. I repeat, from the search space, we are looking for those solutions which are not dominated by any other solution. If we consider x7, can it be considered optimal? Because there are two solutions which dominates x7. So x7 is not optimal. What about x11? Is it optimal? x11, you see that there are many solutions, x1, x2, x3, x4, all these solutions dominate x11. So x11 is also not optimal. But what about suppose x5? Is there any solution in the search space which dominates x5? You see that there will be, I will say that some solution will be dominated by at least one solution if there is at least one solution within that box. I mean that within this box. Then only x5 will be dominated. But in this case, there is no solution within this box. So x5 is not dominated by any other solution in the search space. So basically, all these solutions on this line, which are at the edge of the search space, all these solutions are not dominated by any other solution in the search space. But the solutions which are inside, they are having at least one domination by at least one solution. Right? So, optimality definition in case of multi-objective optimization problem is that we are looking for the non-dominated set of solutions from the complete search space. This non-dominated set of solutions will be called as optimal solutions or Pareto optimal solutions. Pareto is the name of the scientist who actually first dealt with this kind of problems. So it is called the Pareto optimal solutions. And so for a multi-objective optimization problem, we are actually searching for Pareto optimal solutions. And most of the cases, these Pareto optimal solutions will not be a single solution rather than a set of solutions like this. So this is also called this front. This line is called a Pareto optimal front, or Pareto front in short. So any solutions on this line will be a Pareto optimal solution. Hope this Pareto optimality definition is clear. So we are basically looking for the set of Pareto optimal solutions. But the problem is that due to many things, it might happen that you might not be getting all the solutions on the Pareto optimal. Ideally, you should get all the solutions on the Pareto optimal front. But due to the complexity of the problems, due to the size of the problems, due to the constraints, your algorithm for multi-objective optimization might not reach the actual Pareto optimal front. So you are basically trying to approximate the Pareto optimal front. As many solutions, or you want to go as close as possible to the Pareto optimal front. Let me test whether you have understood. So suppose this is your objective space. This blue color shape is your objective space. And suppose both F1 and F2 are minimization objective. Can you tell me that where will be the Pareto front? This is very similar to the previous diagram. Both are minimization. So we are trying to minimize F1, we are trying to minimize F2. So the Pareto optimal front will be on this edge, like this. So this will be the Pareto optimal front. Now what will happen if F1 is minimization and F2 is maximization? Then where will be the Pareto optimal front? We are trying to minimize F1 and we are trying to maximize F2. So here, anywhere else, you see the picture, actually this part and as well as this part. Why this part? Because these solutions will be also non-dominated. You see that any solution you consider and compare with any other solution inside, these solutions will have the better value in F2. So that any solutions on this line will not be dominated solutions. So these will also come in the Pareto front. So Pareto front need not to be continuous. That's why I am just showing this example. The Pareto front need not to be continuous. So it can be like this. Now what will happen in F1 is maximization and F2 is minimization? Where will be the Pareto front? Obviously this will be here, right? F2 is minimization and F1 is maximization. So Pareto front will be here, like this. And if both are maximization objectives, then it will be towards the northeast part, right? So like this. So it depends whether you are dealing with minimization objectives or maximization objectives. It will determine where the Pareto optimal front will actually exist. But for simplicity of implementation, to facilitate the implementation of the problem, what we most of the cases do, that we convert all the objective functions into one type. So you generally have a mix of minimization and maximization objectives. But what you can do is you can convert all the maximization objectives into minimization. How? Suppose we want to say maximize the mileage. So how it can be converted into a minimization objective? You can put a negative before that. So you are maximizing the mileage means you are minimizing the negative mileage. So that's why, or also you can take the reciprocal. So anything you can do. So this way, all the objective functions are generally converted into one type to make it easy for the implementation purpose. So if we compare the single objective versus multi-objective optimization, so here we have multiple conflicting objectives as opposed to the single one. And we may also have the multiple optimum solutions. Than a single solution, we have generally a set of Pareto optimal solutions. Two goals. Unlike the single objective case, the only goal is to reach that objective function, the minimum or optimum value of the objective function. But here two goals. One is progressing towards the Pareto front as much as possible. So we call it convergence goal. The algorithm that are trying to solve the multi-objective optimization problem is trying to come as close as possible to the Pareto front and maintaining a diverse set of solutions of the non-dominated front or Pareto front. Diverse set of solutions is important because it might happen that some algorithm is very good in convergence. That I just give you the example. For in this case, suppose some algorithm comes very close to this part of the front, but is not able to capture this part of the front. So maintaining the diversity is important. We are basically trying to capture the complete range of the Pareto optimal solutions. Some algorithms might be good at concentrating in a particular range of the Pareto optimal front because they are not maintaining enough diversity in the front. Okay? So we have to also maintain the diversity. We have to consider the convergence. We have to give consideration to the convergence. We want to come as close as possible to the Pareto optimal front, as well as we want to capture the complete front. That is the total range of the Pareto optimal solutions. So diversity maintenance is also very important. And sometimes these two tasks are orthogonal. Orthogonal means they are contradictory sometimes, that some algorithms will be good in convergence, but they are not good in maintaining diversity. They will miss some parts of the Pareto front. And other algorithms might be maintaining diversity well, but they are not coming as very close to the Pareto front, actual Pareto front. And we have to deal with two such spaces, as we have already mentioned, that one is the decision space and another is the objective space. And the difficult problem is that the proximity or closeness in one space does not guarantee the closeness in the other space. That means two solutions, two curves are very similar in decision space, but they might not be very similar in the objective space as well. And the search will be definitely performed in the decision space because we are trying to find the best combination of the decision variables. And no artificial fix-ups means there were some earlier approaches where we tried to combine the multiple objective functions into single one by summing the objective values using some weights, something like that, so that a multi-objective optimization problem is converted into a single objective problem, and then use the traditional single objective optimization algorithms to solve that problem. But that has different problems again. That has several issues. For example, if you want two objective function, one is the distance, another is the cost. How can you sum up these two? There is no meaning of just summing a distance with a cost. And also, you might not be aware of the relative importance of the different objective functions. So, and also the different objective functions might be of different range, having different range values. Like some objective functions varies between 0 to 1. Some objective function has very high range, minus 500 to plus 500. So just adding them up together what will happen that for some objective function will have more effect on the combined objective value. So what you have to actually do that you have to optimize multiple objective functions simultaneously, not combining them together. They have to be individually optimized but simultaneously. I will give you an example of a well-known multi-objective optimization. So genetic algorithms are search and optimization algorithms, a solution which optimize certain objective functions. So this is the classical genetic algorithm that deals with only one objective function. Now this is how this is inspired from the Darwin's evolution principle. So for that we have to first understand what Darwin actually said in his evolution theory, evolution of species. So Darwin said like this in very simple terms. Darwin said like this, that initially in the earth there are only very primitive small creatures. So I am talking about the evolution of human beings, suppose. We started with very primitive creatures, and this set of individuals initially they actually tries to cope with the nature. They tried to adapt the nature. They fight with each other for the food and safety. And finally, only the fittest ones survive. So you probably have heard that fittest terminology, that survival of the fittest. The Darwin's terminology, the survival of the fittest. So only the fittest individuals survive. Only the better individuals survive. Then these better individuals, they take part in mating. They take part in mating to give birth of next generation children. So this set of children, what we call as the new generation. We started from initial generation, now we have a new generation of individuals. This process continues. Again, these children will try to survive, we try to cope with the nature, we try to adapt the nature, will fight among each other, and only the fittest ones will survive. And again they will take part in mating, give birth to the next generation. So this loop will continue, this loop continues. So generation after generation, what we can expect? We can expect a better set of individuals, because only the better individuals are taking part in mating and giving birth to the children, so their children are expected to be better. So this is going to be better and better, and thus the evolution happens. And finally, at the current point of time, we will find the best individual that will be our final solution. Now, how this can motivate an optimization algorithm? In an optimization algorithm, what we have? We have again a search space. Generally, we start with picking up some random solutions from the search space. Picking up some random cars, suppose, in the search space. And suppose we are dealing with only one objective function, that is the cost of the car. So picking up some random cars, so this is my initial generation of individual solutions. Then we compare these cars with respect to the objective functions. So when we are talking about the survival of the fittest, so how this fitness is defined in this case for this problem? When we are comparing two solutions, which solution will be more fit? In what terms? Obviously in term of the objective function value. Objective function is cost. So when we are comparing two solutions, the lower cost means a better individual. So out of the initial set of solutions, we select the best set of solutions, or at least some better set of solutions. So this is called the natural selection and the survival of the fittest. There are some methods or techniques how to select, but the idea is that from the initial set of solutions, we are picking up some new set of solutions which are supposed to be better in terms of objective functions. So these solutions will then be combined with each other, giving mating is simulated. Mating is simulated in terms of crossover. We call it crossover. Now for helping the crossover part, what we actually do, that we represent each solution in a form of a string. Each solution means the decision variables, the length of the card, the width of the card, the height of the card. Suppose these are the three decision variables. So we can convert them into binary and join them together, so that will give you a binary string. So we call it a chromosome, just to mimic the biological inspiration. We call it a chromosome. It is nothing but a string. Now this is how a solution is represented. And then we perform a selection operation. There is some selection mechanism. One simple selection mechanism is called the binary tournament selection. What is binary tournament selection? So suppose we have the initial generation of solutions. In binary tournament, what we do, we randomly pick up two solutions and then choose the better one in terms of the objective function value and put it in a pool of mating. We call it a mating pool. That means the pool of parents who will take part in mating for the next generation. Okay? So we randomly pick up two solutions, put the better one in the mating pool. Again we pick up randomly two solutions and put the better one in the mating pool. So this way we do it several times to fill up the mating pool of solutions. Then we perform a crossover among the solutions in the mating pool. How the crossover is performed? We again randomly pick up two solutions from the mating pool and exchange their parts. Exchange the parts. Suppose this is one solution, this is another solution. We choose a random crossover point and then we get two children by choosing, for example, for these children the first part comes from the first parent and the second part comes from the second parent. And for this child, the first part comes from the second parent and the second part comes from the first parent. So this way by crossover we get two new solutions, two new strings, two new solutions. Then we perform a mutation operation. Mutation means, Darwin said that mutation is a very rare operation that happens during the evolution process. Very rare occurrence, only a small change in the individual's chromosomes. So here the small change is basically, suppose among the total length of the chromosomes, 2 bits are mutated. These bits and this bit is mutated. So here mutation is only possibility is that if the bit was 1, it is converted into 0. If the bit is 0, it is just flip. This is the flipping. There are other types of mutation. I am not going into that, but this is that way of mutation. So this way every child is mutated with very low probability. So this will give birth. So this is the whole process. You see that this is the initial population. We first evaluate the fitness values, that is the objective function values for each solution. Then there we test some termination criteria is there. If the termination criteria is not met, then we go for the selection, that binary tournament selection, suppose. Selection will create the mating pool. From the mating pool, we perform crossover and then mutation. So that will give you the next generation population. So again, we compute the fitness values of these next generation individuals, and we continue this loop until some termination criteria may be met. For termination criteria, it might be you just run certain number of iterations, suppose 100 iterations or 200 iterations. And what will be the final solution? That is the solution that gives the best value of the objective function in the final generation. So that will be the optimum solution. Optimum, I'm not saying optimum, but it is approximation of the optimum solution, because it is not guaranteed. When the search space is sufficiently very large or infinitely large, you might not get the optimum solution always. But genetic algorithm, the good thing is that it guarantees to take you a near optimal solution at least. There are some theoretical proofs, I'm not going into that. So this is the idea of genetic algorithm, how it works. So obviously you see that this is a randomized search and optimization technique. Now this is the classical genetic algorithm where we have to deal with only single objective function. Now can you tell me that if you have to deal with multiple objective functions, where in this loop that will affect actually? Will it affect the initial population generation? Generally it is generated randomly. We generate some random binary strings. So that will be also similarly done in the multi-objective case. Fitness evaluation, will it be different? A little bit different, because in single objective case, we have to evaluate one objective function value. Here we have to evaluate each and every objective function value you are going to optimize. 2, 3 objective functions, all of them have to be evaluated for each individual, right? Termination criteria, if it is just the number of iterations, it is also not going to differ. Selection, so here is the main difference, right? Because here the selection you cannot simply apply binary tournament. Binary tournament you can apply, but how? I mean, you are picking up 2 solutions, there will be 3 possibilities, like in the first 2 cases, if one solution dominates the other one, then it is fine. You just pick that solution and put it in the mating pool. Now what will happen if these 2 solutions are non-dominating? They are non-dominating. Then which solution to pick up? So that you have to answer. So the selection is the main thing that will differ from single objective case into multi-objective case. And then crossover and mutation will be again same, because they don't have to do anything with the objective functions. We are just operations on the strings. So we are basically looking for this selection. Let us see that how we can perform the selection, or how we can modify the binary tournament to use it in case of multi-objective optimization. Now there are over the years, lot of different multi-objective optimization algorithms have been proposed based on genetic algorithms. So obviously it is not possible to discuss all of them, but I will discuss one of the very popular genetic algorithm, multi-objective genetic algorithm, which is called the non-dominated sorting genetic algorithm, and its second version, NSGA2. But NSGA3 has also come, but that generally deals with many objective. So now the research is going on many objective optimization. Many and multi, the difference is that for multi we are dealing with only 2, 3, 4 objective functions. For many we might have 100 objective functions. So anyway, let us see that how this NSGA2 is actually implemented. So NSGA2 was proposed it is almost more than 20 years now. but still very, very popular algorithm. People are still using that. And it was proposed by Professor Kalyan Mohadev. That time he was in IIT Kanpur, but now he has moved to US. Now this non-dominated sorting has two main concepts. You see that I said that in multi-objective optimization, we have two goals basically. One is the convergence, that means come as close as possible to the Pareto optimal front. Another is the maintaining the diversity to capture the complete front. So the first objective or first goal is actually taken care of by this non-dominated sorting. I will give an example. What is this? This is nothing but ranking the solutions in the search space, or in the set of solutions you are considering. We try to rank the solutions in terms of their domination status. And the diversity, for diversity maintenance, they provided a concept called crowding distance. I will again discuss that. And elitism is also, because you see that as GA is a randomized approach, so what might happen that at certain point of time, suppose you have got a very good individual solution, but due to randomness of crossover, mutation, selection, you might lose that solution. So that this kind of thing does not happen, we want to preserve the good solutions. We use a technique called elitism. I will discuss that also. So this is the time complexity, I am not going into that. So first of all, what is non-dominated sorting? Suppose this is my initial population of solutions. This is the objective space. We have two objective functions, suppose F1 and F2, both are minimization objectives. And these are the solutions in the objective space, initial solutions, initial population of solutions. Now non-dominated sorting means, first what we do, we find out that what are the non-dominated set of solutions in this set of solutions. Which solutions are non-dominated? You can easily do that. You can just write an algorithm. You take the first solution. You compare the first solution with every other solution. As soon as you get another solution which dominates this first solution, you stop. You don't have to see the other ones, because one domination is sufficient to discard that solution. So if you check with the other solutions, if you see that the first solution is not dominated by any other solutions, so that will be part of the non-dominated set. You repeat the same thing for the second solution. You compare with the other solutions. If it is not dominated by any other solutions, then this will be also part of non-dominated set, and so on. So this way, from these solutions, we first find out that which solutions are non-dominated. So these solutions will be non-dominated. As we have already seen, both are minimization objectives. So in this space, the non-dominated front will be on this edge, right? So these solutions are non-dominated solutions. So we call these solutions as the rank 1 solutions. So these are the best set of solutions, no doubt. Rank 1 solutions. Next, what we do, we temporarily remove these rank 1 solutions. And from the rest of the solutions, we again find which are the non-dominated. So they will be called as rank 2 solutions. Right? Hope the procedure is clear. So these are the rank 2 solutions. We repeat the same process. We remove the rank 2 solutions again. From the rest of the solutions, again find which are non-dominated. So these will be rank 3 solutions. We continue with this. How long we will continue? As long as or until every solution gets some rank. We will continue until every solution gets some rank. So these are rank 3, then rank 4, then rank 5, then rank 6, then rank 7. So all the solutions have got some rank. So this procedure is called non-dominated sorting. From here the name came, that non-dominated sorting genetic algorithm. So this is NS or non-dominated sorting. So basic idea is that we are ranking the solution based on the non-domination status. Their non-domination status, right? So next thing is that crowding distance. Crowding distance is that, so now suppose given this situation, if we want to perform a binary tournament selection, how we do that? We pick up two random solutions. If the two solutions are coming from two different ranks, then our choice is easy. Suppose one solution is coming from rank 2, another solution is coming from rank 5. Which solution you choose? Rank 2 solution. But the problem happens when both the solutions, because you are picking up randomly, it might happen both are coming from the same rank. Then which solution you should choose? One possibility is you choose one randomly. But that will not guarantee the convergence. Actually that NSGA, the first version of NSGA did it like that way. That when two solutions coming from the same rank, they choose the randomly one. But in NSGA 2, to maintain the diversity, here another approach is proposed, which is called the crowding distance based approach. It is like this, that first of all, what is crowding distance? Let me define. Suppose this is one rank of solutions, okay? One particular front. Now for every solution, we compute a crowding distance. Crowding distance actually, it tries to capture that where the solution is situated, how crowded that region is in the front, how crowded by other solutions, whether there are any other solution nearby. How that is computed, you see. So first of all, suppose I want to compute the crowding distance of this solution. So this is done like this way. We take the the solution that comes immediately before this solution, I minus 1, and immediately next solution, I plus 1. Then we draw a box, virtually, definitely, we draw a box around it. And the perimeter of this box, or also you can consider the area of this box, will be your crowding distance for this solution. And it is easy to compute the perimeter because you can easily compute this value. What is this value? This edge. This is F1 I plus 1. F1 of solution I plus 1 minus F1 of solution I minus 1, right? This part. And this edge will be F2 I minus 1 minus F2 I plus 1. So you can get this, the value of these two edges, length of these two edges, so you can compute the perimeter or area of this box. Similarly, suppose you want to find the crowding distance of this solution. So that will be the area of this box. So it is obviously much smaller area than this one. And now what this area or what this crowding distance actually indicate? What it means if some solution has a larger crowding distance? It means that the solution in this front, the solution is situated in relatively less crowded region. Less crowded region means that there are no other solutions nearby. But for this solution, it is situated in a more crowded region. There are many other solutions nearby. So suppose you have picked up two solutions randomly. Suppose this solution have been picked up and this solution has been picked up for binary tournament. So they are coming from the same rank. So if they are coming from the same rank, which solution you will prefer? Can you tell me that solution I or solution I plus 2? Which one you will prefer? Solution I. Why? Because your goal is to capture the complete Pareto front. If you are going to lose this solution, as there are not many solutions nearby, you might lose that part of the Pareto front. But if you can afford to lose these solutions because there are some other solutions nearby to capture that part of the front. So when we are comparing two solutions from the same rank, I will choose the solution with the higher crowding distance value. Now what about the two-end solution, the solution that has the minimum value for F2 and the minimum value for F1? We cannot afford to lose these two solutions because these are the two ends of the Pareto front or non-dominated front. So what we do, we give infinite values of crowding distance to these solutions, so that whenever one of these solutions is compared with some other solutions in between, always the edge solutions will be chosen. So with crowding distance, we can now modify the binary tournament. It is now easy. So binary tournament, so we pick up two random solutions, A and B. First we check what are their ranks. If they are coming from two different ranks, we choose the solution having the lower rank, right? But if they are coming from the same rank, we choose the solution with higher crowding distance. So when we modify the binary tournament in this way, this is called crowded binary tournament selection. So instead of binary tournament, simple binary tournament, here we use crowded binary tournament selection. Right? So how much time I have? Okay. I don't think there will be enough time for application. Anyway. So let us see that now the complete loop of the NSGA2. So we start with the initial population again, like single objective case. Then we perform non-dominated sorting. That is, we simply rank the solutions using non-dominated, rank 1 solutions, rank 2 solutions, rank 3 solutions, and so on. Then we perform selection, that is the crowded binary tournament selection, to create a mating pool of solutions. Then we perform a crossover and mutation. So this is the next generation solutions, children solutions. So we call it, this is the parent population, this is the child population. So this child population will now become the parent population for the next generation, same iteration. Now, as I already mentioned that due to randomness we might lose some good solutions. To avoid that, what we do, we use a technique called elitism just to preserve the elite solution. Elite means the very good solutions. So for that, what we do, we combine this parent and child population together. And then this combined population is, on this we apply non-dominated sorting. On this combined population we apply non-dominated sorting. And then from these, because suppose we started with population size P, now here we have population size 2P, right? But we still want population size P. So from these combined solutions, we pick up P-based solutions. So we pick up, suppose, all rank 1 solutions, all rank 2 solutions, and some of the rank 3 solutions, because if we pick all the rank 3 solutions, it will exceed size P. Then can you say that which solutions from rank 3 will be chosen? Based on what? Crowding distance. So as all these solutions come from the same rank, we sort them in descending order of the crowding distance and choose from the top the required number of solutions to give you the P number of solutions. So this set will be considered for the next generation. So we have already performed the non-dominated sorting, so this part will not be needed. We continued from this part. Directly we can start with the selection process, right? So again, mating pool, crossover mutation, elitism, and this loop will continue for a certain number of generations. And what will be my final solutions? What will be your output? So in the final generation, what we have? In case of single objective case, my output is the best solution in the final generation. Here, the rank 1 solutions of the final generation, right? So that will give you the approximate Pareto front. Why am I saying approximate? Because it is not guaranteed that it will always capture all the solutions of the Pareto front, but the basic goal of any multi-objective optimization algorithm is to capture or approximate the Pareto front as close as possible and the complete range of the Pareto front as much as possible. Okay? So this is your NSGA2, and there are many benchmark multi-objective optimization problems. All of them are basically 2 objectives. I just quickly go through the Pareto front of these solutions. So this is the first problem. So you see that this is a very convex shape Pareto front like this. This is a concave shape Pareto front. Now this is a non-continuous Pareto front, I said. So this is again a discontinuous Pareto front. Because actually this part of the search space is something like maybe something like this. So that's why that part is not coming. So it is continue from this part again. So this is a good example of non-contiguous Pareto front. Because actually the line is like this. So these parts are obviously not coming in the Pareto front. Okay. So these are some example. Maybe I can get another 10 minutes, right? 10 to 15 minutes. So just quickly go through that one of the applications that we did in bioinformatics problem. So the application is actually that on identifying the gene markers. So let me quickly explain what is this. So you see that we all know that our body is made up of billions of cells, right? And inside each cell we have a nucleus, and within the nucleus we have a set of chromosomes. Real chromosomes, not that artificial chromosomes. These are the real chromosomes. These chromosomes are made of a molecule, all of us know the name, that is called DNA. This chromosome is a nucleic acid molecule that is called the DNA, D-riboxynucleic acid. Now, these DNA, within a chromosome, there are some parts. So chromosomes are like strings, okay? So DNA molecules are nothing but a string of, if you just convert it into a computerized string, it is nothing but a string of 4 characters, A, T, G, and C. Maybe in school level we studied that, A, T, G, and C, adenine, thymine, guanine, and cytosine. So this is a string of A, T, G, C, and in any particular order. This order is important, that is controlling our life. But in this ATGC string, not every part is actually a code. Some parts are the codes. Some parts has the important information about how the cells perform. These parts, do you know what these parts are called? We all know actually the name. These are called the genes. Okay? So in the chromosome, which is made up of DNA, some fragments, some sections are the genes. But these genes are inactive. Inactive means these genes cannot function themselves. It is just like, in computational terminology, it is just like programs sitting in the hard disk. The program has the code, what to perform, but they cannot perform by themselves until they are executed, right? Until a program becomes a process, then the function starts. The program comes to the main memory and then the function starts. So similarly, genes are the codes. They cannot function themselves. So these genes are then, the functional units are actually called the proteins. So from the gene, there is a process called the central dogma of molecular biology, which is a two-step process. From the genes, first they are converted into mRNA or messenger RNAs, and from RNA, the proteins are created. Now these proteins are nothing but a sequence of amino acids. Okay? So genes are sequence of nucleic acid, ATGC, and the proteins are sequence of amino acids. So these proteins are actually the functional units of the cells. All the functions within the cells are performed by some proteins. Either individually or different proteins interact with each other and so on. Now the good thing is that almost all the cells of our body have the same set of genes. So they are capable of producing same set of proteins. So different proteins have different functions. Some proteins create the membranes of some items. Some proteins may carry some molecule from one part to another part, and so on. So different proteins are different. So every hormone is a protein, right? So you know hemoglobin, it is a protein. It is carrying oxygen molecule from through our bloods. Now the thing is that almost all the cells have the same set of genes. So we can say that all of them have the same code. All the cells have the same code. Then why our lung cells perform differently than our skin cells? Why our heart cells perform differently from our kidney cells? Any idea? They have the same code, same program, or set of programs. Each gene is a program, so we have a set of program. But their functionalities are different. Why? Because not all genes are creating protein in every type of cells. Not the same set of genes are creating proteins in every type of cells. The subset of genes which are creating proteins in our lung cells are different from the subset of genes which are creating proteins in our skin cells. So when a gene starts creating protein, that means when a gene is converted into an RNA, DNA is converted into an RNA, we call the gene is expressed. So not all the genes are expressed in every type of cell. So this phenomena is called differential gene expression. And that is also true when we are, suppose, dealing with some patients in medical treatment. Suppose we are dealing with the cancer patients, a specific type of cancer. Suppose the genes which are normally not expressed in our, suppose we are dealing with lung cancer, it might happen that the genes which are normally not expressed in our lung cells, but when cancer happens due to some mutation, those genes starts creating proteins. Those genes become active and gets expressed. So normally the genes are not expressing, not creating proteins, but when cancer happens, the gene starts creating proteins. So these genes are called the oncogenes. And there is a technology called microarray technology by which we can measure the amount of expression, gene expression. Amount is measured in terms of how much RNA a gene is producing. So the amount of RNA a gene is producing in a normal cell might be defined from the amount of RNA the gene is producing in a cancerous cell. Right? So this is called the differential gene expression. So these genes, this is not true for all the genes. Some genes are unaltered. That means throughout either normal cells or cancer cells, they have the same expression pattern. But there are some genes which are not expressed much in the normal cells, but expressed highly in the cancerous cells. So these genes are called oncogenes. From the oncology, these genes are called oncogenes. And there are also other types of genes like which are highly expressed in normal cells, but they are not expressed in the cancerous cells. So these genes are called tumor suppressor genes, because when these genes were active, that there was no tumor. When the genes get switched off, a tumor grows. So normally their activity is suppressing the tumor. So these are called the tumor suppressor genes. So these oncogenes and tumor suppressor genes, these are called the genetic markers, marker genes, because these genes will tell you that whether a patient has cancer or not. Suppose you are going for diagnosis, you know that this particular gene is oncogene. So you just look for what is the expression value of that gene in that patient. If that is a very high expression, it is supposed to be a malignant patient. Right? If the expression is not that much, then maybe it is a normal tissue. That the tumor might be a benign tumor. It is not malignant tumor. Okay? So the idea is that we have to find out which genes are marker genes. So if you have so the microarray data comes to us in this way. So it is nothing but a matrix where suppose the columns represent genes and the rows represent the different tissue samples from different patients, might be different patients. So now there are some patients which are already known to be normal patients, they don't have malignancy, and there are some patients which are having malignancies, the cancerous patients. And these are the expression values, normalized expression values of the genes. And these colors actually gives you the amount of expression. The more reddish is the color means that higher is the expression. We just colorify the values just for the visualization purpose, right? So this is called the heat map. Heat map means because the more red means the high expression, more greenish means the low expression, and blackish means the middle level expression. So the thing that we are going to do that, so I already explained what is a gene marker. So gene marker identification can be posed as a feature selection problem. That means in this data set, so we call these columns as the features because each sample is defined in terms of these feature values. And we are looking for those features out of all these. These data sets are fairly large. That means suppose 10,000 genes, 20,000 genes in the columns. But the number of samples is very small normally, 100 samples, 150 samples and so on. So this problem is called the curse of dimensionality. When your data set is like you have too many dimensions, so we have problems there. So here the goal is that you have to identify those columns, those genes, which have suppose very small, so almost all the samples under the normal samples, all of them are greenish. And all the samples in the cancerous samples, all of them are reddish samples. That means low expression value in the normal samples and high expression value in the cancerous samples. So that gene can be considered as a marker gene. Or the opposite thing, that means all the samples are red here and all the samples, all the values are greenish here. So those will be the tumor suppressor genes. So both cancer, the oncogenes and the tumor suppressor genes. So these are the marker genes that we want to find. So we want to find such marker genes so that using those genes we can easily, if a new sample comes, suppose, for which we don't know whether that is a normal sample or malignant sample, we can identify that sample by just considering what is the expression value, and if it is for a marker gene, and accordingly we can classify the sample as to be either malignant sample or normal sample. So it can help in diagnosis. And also these marker genes can be also targeted if new therapeutic approach. That means due to some mutation the gene has become active due to cancer, we can target that gene so that we can use some kind of medicine or some kind of chemo to just suppress that gene, or to make that gene inactive again, so that it can help to treat the cancer. So that actually posed as a feature selection problem. It is like this, that we are given the original data sets with the all genes, and we are basically trying to select a subset of genes, evaluate that selected subset based on certain criteria, and this process continues until we get a final subset of gene markers. Now this is actually, we use the NSJ2 for doing this. So here a solution means a subset of genes which are marker genes. So we can represent a subset of genes by means of a string, as I said that each solution has to be represented as a string. So we can represent a subset. Suppose there are total 10 number of genes. So we can create a binary string of length 10. So where the values of the string is 1, we can select those genes as the subset. Okay, that is the selected subset. So this way we can generate initially a random set of binary strings to represent the different possible subsets, and then we evaluate each string using objective functions. Now what are the objective functions here? We use two objective functions actually. So this is the process. So suppose I am evaluating one chromosome, that is the selected subset is 1, 4, 6, 9, 10. So we project the whole data set on those genes only. We just discard all other genes, and then we apply a classifier, because we already know these samples are labeled samples, right? Because we already know that these are the normal samples, these are the cancerous samples. So we can apply a classifier using some cross-validation technique. I don't know that whether you are familiar with these terms. Maybe the classifier is just a model that can predict the class levels of the, it is first trained by the labeled samples, and then they can predict the class levels of the unknown samples. So we can use the neural network classifier here to classify or to predict the labels of the samples. And these predicted labels are then compared with the true labels. So this prediction is done only based on these genes. So these predicted labels are compared with the true labels, because the true label was known, and we just compute what is the accuracy. What is the percentage of the accuracy? That out of suppose 6 samples, suppose you have predicted correctly 4 samples. Their levels have been predicted 4 samples. So your accuracy is 4 by 6 and converted into percentage. So suppose we get 70% accuracy. So that is one objective function. And another objective function we use, that is the number of selected genes, number of features, because we want to find the smallest subset of genes that is capable of distinguishing the two types of samples. So the number of features is to be minimized and this accuracy is to be maximized. So two objective functions. We are trying to maximize the accuracy we are trying to minimize the number of features. That means number of features means number of ones in the string that we want to minimize. So we can again convert it into one type of objective, just putting a negative in one of the objectives. And then we applied NSGA2 to find out the possible set of solutions. Possible setups, I just give some examples. So this is the normal NSGA2 has been used, single point crossover mechanism and bit flip mutation. And these are the parameters. We ran it for 100 times, 100 generations, population sizes. So these parameters are issue actually, so because there are some trial and error method actually you have to do. And here are some example results. You see that this is one real life data set of colon tumor. So this is the whole data set. Here actually we have transposed the data set. Here the rows represent the genes and the columns represent the samples. So these are the selected set of genes. You see that within this blue box, this is the selected set of genes by our method. So can you tell me that what these genes are actually? These two genes. So this is one gene. So each row is one gene. So you see that these two genes have lower expression value in the tumor samples and high expression value in the normal samples. So these are basically tumor suppressor genes. And these genes are on average higher expression value in the tumor samples and smaller expression value in the normal samples. So these are the oncogenes. Okay? So these way, and most of the other genes are random. I mean, there is no particular pattern that here low, here high, no particular pattern. So these genes are kind of noise. They will not help you distinguishing the type of the sample. But these genes can help you distinguish the type of the samples, right? So these are the marker genes, and we repeat it for another data set, so leukemia data sets. So here there are two types of leukemia actually, ALL and AML. Again, these are the marker genes, very clearly visible, right? So these genes are red, these genes and green in the other kind of samples, and these genes are green in the one kind of samples and the red in the other kind of samples. These data sets consist of too much of noise, okay? Due to the experimental errors, there are too much of noise. So there will be certain cases like within the red, there will be certain number of greens, but that is okay because of there might be, so we are just dealing with that what the most of the genes are saying. So that's the idea. And these are the some of the public real life microarray data sets. You can, if you want, you can download from them, from these sites. And for the NSGA2 code, if you want, you can visit this site, Kanpur Genetic Algorithm Laboratory, or Kangal in short. There you can get the code for the multi-objective genetic algorithm, or NSGA2 code you can get. And also, generally the computer science people will want to write their own code, because we prefer to write our own code, because if you want to modify the code in certain at certain points, then it is always better to deal with my own code than just understanding others' code and then modifying. It will be difficult, right? So I think that I missed the conclusion side anyway. So these are the some of the reference of our papers where we applied this multi-objective optimization. There are many real-life domains you can actually apply. Almost most of the real-life optimization problems are multi-objective in nature, and we applied in image processing also. We applied in there are other bioinformatics problems there also we applied, and there are some facility location problems. Recently we applied multi-objective optimization. So this is one of the books that we wrote. this was actually outcome of my PhD thesis that time. So it is more than 10 years back this book was published. So any question? And yeah, so if you have any questions, I will be happy to answer. Or also if you want to contact later, you can drop me a mail here. And you can look into my site. There some of my papers are available in PDF versions. If you want, you can just go through that papers. Yeah. Yeah. Yeah, I'll come to that. These ones, right? So these ones nothing but some benchmark optimization problem, multi-objective optimization problems. So these are the two objective function values, okay? So F1, so this is n equal to 1 means that number of decision variables is 1, only x, okay? One objective function is x square, another objective function is x minus 2 square. And that gives you a Pareto front like this, okay? Similarly, for the more complex problems are there. So these are designed problems, okay? For which the true Pareto front is known. And we just then apply a multi-objective genetic algorithm and to compare the output with the true Pareto front, how it is performing. Okay. I think I have given these slides to madam, so you can collect it from there. Hope you have got at least some idea of multi-objective optimization.